Welcome back, friends. This is another video regarding development biology from Shomu's biology, and in this video lecture we are going to talk about the limb development in vertebrates. So I'm going to make this video in the PowerPoint presentation manner because there are some pictures I want you to show. So let's look at it. To understand the vertebrate limb development, you need to first know about the structure of vertebrate limb a little bit. So let me pick a color here. And I can tell you, like, all of us know how the limbs generally look alike. Like if you talk about humans, in that case this is a hand. If we talk about like something like this is a kind of structure that we always see. So if you look at a limb structure, it's like an extension from our body, right? And we know the structures which are formed during the development regarding the association and formation of any extension. All those structures are formed from the germ layer ectoderm. Remember that. Okay, ectoderm is responsible to make structures like all this budding, like formation of our eye, limb bud and all these things. So this limb is formed from the limb bud, and limb bud is in turn is formed from the ectoderm. So ectoderm is actually making the structures of a limb possible. So to understand the development of the limb, you also need to know the different axes of a limb, because you know limb can be varying for different vertebrates. For example, in case of humans, the limb can be either hand or leg, but for birds there is only one set of the limb, that is only leg. The rest of the limb, that is the forelimb, is converted to wings, right? So generally when we talk about the limb development, we always talk about bird as an example, because in case of bird, that limb is converted to two different things. One is the leg, another one is the wing. Now the thing is, when the limb development begins, there are two set of limbs that needs to be formed, right? One set is known as the forelimb, another one is known as the hindlimb. So in case of birds, that forelimb is converted to forelimb is converted to wing, and hind limb is converted to leg. Okay, that's the structure. So let's look at here. In this picture you can see that a structure of a limb can have three separate axes. In two-dimensional space you can only visualize two axes, but here in 3D shape you can see three separate axes. One is the proximal to distal. Proximal to distal means the place from where the limb is linked to your body, that is the proximal, and distal is at the end of your fingernails. Okay, this is proximal to distal, PD axis. The second type of axis, if you look at a hand in a 2D space here, dorsal and a ventral. Dorsal side facing the top regions, and ventral is at the region of your palm. And the third axis is posterior anterior. Posterior is where you have the pinky hand finger, and anterior where you have the thumb. So this is a type of three separate axes that are always present. Okay. So now if you look at here the picture in the left hand side, here I am going to tell you the important idea about how the limb is developed. Now as you know what is limb and what are the different axes of the limb, and we know whenever there are different axes of any body, then we need to have a body plan, so that according to the plan we can establish the structure of that organ, right? So in this case there are two things that you need to understand. First thing is idea of induction, and second thing is development, and third is the maintenance. These are the three situations that you need to understand. Induction is a very common phenomena during development, where one set of cell, which is a donor cell, starts secreting some chemical components which creates a signal transduction, and after result of the signal transduction a specific organ is a structure of an organ is achieved. Induction says that the cell secreting the signaling molecules want the neighboring cells to find and form same type of structure like the cell where the signaling molecules are released. Okay, so here we will see there are few cells which are secreting some chemical components, some signaling molecules. Now the important fact about induction is, let's say there are So now if I draw a situation to explain the induction, it's a very important phenomenon. Let's look at it. So let's say there are few set of cells adjacent to each other, adjoining to each other, okay, and all this set of cells that are present with each other, and they also have a nearby cell, and this nearby cell is a secretory cell, okay, containing all the chemical factors, and starts secreting the chemical factors which are acting as a signaling molecule on the nearby cell, and that will allow the nearby cell to be converted to a specific organ structure, the desired organ structure, okay. Now the beauty about induction is whenever this neighboring cell, this signaling cell releases the signaling molecule, the neighboring cell receiving the signaling molecule may behave differently. Let's say one side of the cell, one type of the cell here, they are converted to a specific structure, while the other cells receiving the same signaling molecule may form a different structure, okay. And also if the signaling molecule is altered, if the signaling molecule is changed, that is also going to change the response and the type of structure that they are going to form. So in case of limb development, we will see there are major two type of signaling molecule that will play a vital role in finding and identifying the structure and instructing the structure to be formed, either forelimb or hindlimb, okay. Because in case of human also we have forelimb and hindlimb, but let's say in case of birds, the conversion of forelimb is into the wings, that is a separate job, but at the very beginning the induction process is very similar, okay. And to understand the signaling process, we have two things. One is the signaling molecule that we need to know, the signaling cell, and where exactly they are acting. So there are actually two places which are really important for the functionality and formation of the limb. One is the AER, another one is the ZPA. AER known as apical ectodermal ridge, a structure modified version of the ectodermal region. This is the structure which is mostly found towards the distal end of the ectodermal region of initiation, from where the limb bud start to generate, okay. So AER is an important place where all the signaling molecules will work. Now what are those signaling molecules? We will see in the next slide, but here we are looking at where exactly the signaling taken place. That is, one is AER. The other one, ZPA, zone of polarizing activity. This is a part of mesenchyme. Now this zone of polarizing activity is really important for distinguishing between the posterior anterior axis, and AER plays a vital role in finding the dorsal ventral axis and proximal distal axis. So AER responsible for both this type, proximal distal and dorsal ventral, while ZPA is for posterior anterior axis determination. And without determining the axis, the cells will not get the signal to produce a proper structure, because the structure of hand always require all these axes to be properly formed. Otherwise there is no meaning of having a vertebrate, a limb in a vertebrate, okay. So let's look at the next part. Now here you can see an overview of this process. I told you there are two regions, one is the AER, another one is the ZPA, which carries all the cell signaling molecules, and the cells presenting in both these locations upon receiving those cell signaling molecule are subject to change accordingly. But what are those signaling molecules that work? There are three signaling molecules which are really important here. One is TBX5, second one TBX4, and the third one PITX1. Now mainly TBX4 and PITX1 works together for the formation of hindlimb, and TBX5 works alone for the formation of forelimb. TBX is a short form of T-box, and the set of genes out there are known as T-box genes, and these genes ranging from 1 to 28, 30, even more than that, okay. Now specific gene out there are going to code for specific signaling protein known as T-box protein, like TBX5, TBX4, like that. So those presence of TBX5 will guide So the presence of TBX5 will signal a rest of other signaling molecules like FGF and series of FGF8, FGF10 that will ultimately lead to the formation and development of forelimb bud. And when you look at the structure, the process of this limb development takes place at the end of the gastrulation, where the ectodermal structures are properly formed and they start to modify themselves to form AER and ZPA. Now at the end of this process, whenever there is an accumulation of TBX5 that signals the activation of FGF8, FGF10 based signaling cascade, that leads to the formation of limb, and the limb start forming like a bud. So it's a budding that slowly cells start dividing and formation of extension completes. So let's look at the mechanism in details. Here you can see the example of a vertebrate embryo, and you can see here that there are different segments and there are all these different genes out there, T-box genes. And we know when the T-box4 gene is involved, it involved in the hindlimb development. T-box5 gene codes for TBX5 protein involved in the forelimb development. Now in case of chicken, the forelimb bud ultimately converted to wings and hindlimb bud is converted to legs. But if you look at the structure, the number of bones and how they are arranged are very much similar. Little differences are out there, and those differences are modified at the end of the process of development. Here you can see the FGF mediated signaling. I told you that TBX5 and TBX4 both ultimately signals FGF8 or FGF10. Then FGF8 and FGF10 along with another signaling molecule known as SHH leads to the formation of the limb bud. And the formation of limb bud requires constant cell proliferation, because normally the membrane, the ectodermal region is plain, straight like this. So the formation of bud requires a rapid cytoskeleton rearrangement. Okay, so here you can see that this is the first step known as the induction of limb development, where the neighboring cells start releasing the signaling molecule that activates FGF8, and FGF8 induces FGF10. So once FGF10 is activated, FGF10 further will induce the ectodermal cells from where the budding will start. So it's a positive feedback loop always going on between the ectodermal cell and the region of the secreting portion, that is AER or ZPA. Always this process continues there. So by this fashion, when they start secreting this FGF10, so it will allow the cells to proliferate, and then FGF8 induces another signaling molecule, that is SHH, and SHH also further induces the neighboring cells. So sooner what happens, slowly all these cell structures start to modify themselves to form a limb. That's how the limb bud will be generated. So the very beginning was the induction, while the second step is establishment of AER and ZPA signaling centers, and that thing is achieved after the crosstalk between the cells with FGF10 and FGF8. And then finally promotion and outgrowth of those ectodermal cells to form the limb bud, that is when the SHH start inducing the neighboring cells to form those structures. So these are the sequential events of how FGF signaling involves in making a limb bud. Now here we will see all the things that we learned till now, and we connect all these things together at this picture, the true mechanism of this process. We know that TBX5 involved in the formation of forelimb and TBX4 involved in the formation of hindlimb. We also know FGF10 and FGF8 plays a vital role in making the bud of the limb, both in case of forelimb as well as in case of hindlimb. So we know the major signaling molecule, that is FGF10 and FGF8, are same. So how two separate type of limbs are formed? That is the beauty of induction, because I told you at the first place that induction works in a way where a same signaling molecule may lead to two separate effects, or maybe some other signaling molecule lead to the different effect. In this case, we will see both of this activation of FGF10 requires signaling of WNT pathway. WNT signaling can be either WNT2B protein or by WNT8C protein. And if you remember, in the earlier slide also I told you another important thing, that there are this two types of Hox genes that are available. One is this B D, another one is C D. B D if works together, that is the signal for formation of forelimb. C D together, formation of hindlimb. So you can see here that for the WNT pathway, the protein WNT2B is working. So B works for formation of forelimb and WNT8C is working in case of formation of hindlimb. So in either way, this WNT2B when signaling the FGF10, and you see all this signaling are reciprocal. So WNT2B signals FGF10, FGF10 induces WNT2B. So it's always a positive feedback loop, but ultimately starts from TBX5. So leads to the activation of SHH. So altogether FGF10 and SHH start forming the forelimb structure. On the other hand, if you look at here, that TBX4 is activated by this PITX1 gene. Remember, the PITX1 was also present there. So this PITX1 activating TBX4, that further leads to the activation of FGF10 in presence of WNT8C. Now this FGF10 in presence of WNT3A and 8C will signal the limb to be formed as a hindlimb. So if you look at the difference between these two development of forelimb and hindlimb, there isn't much difference. The only difference is the start signaling molecule, that is in case of forelimb it is TBX5, in case of hindlimb it is TBX4. Apart from that, there is only one difference, that is WNT2B presence in case of forelimb, WNT8C is present in case of hindlimb. But actual signaling molecule is always same, that is FGF10 and FGF8. So here the mechanism dictates that ultimately the whole process which differentiate between the forelimb formation and hindlimb formation depends on the release of TBX type. So whether TBOX5 or TBOX4 is going to be activated, because and also the presence of WNT proteins, especially if WNT-B and A, that is a combination to form forelimb, and if it's C and A or C and D, combination of hindlimb. That is the only way they distinguish between forelimb and hindlimb. Now once they form the structure of forelimb and hindlimb, now they need to maintain the structure, because first is the positioning that is done earlier, initiation is also done, that is a part of induction due to the chemical signaling. So the third step is the outgrowth, that is the cellular rearrangement, cytoskeleton rearrangement, formation of a bud, and from that bud cells start dividing and proliferating in presence of FGF8 and FGF10, because both this type are growth factors, and presence of these growth factors the cell will rapidly grow and divide and proliferate to make the ideal structure. Now the third instruction for birds, like a special instruction of converting a forelimb into wing, will come far later of this process. So once everything is achieved, then the further conversion is maintained. Here you can see a list of all this Hox code for all these Hox genes that are available. Now presence of all these Hox genes and the proteins that they make, depending upon those Hox genes, different structures of the bone will be formed. Because if you look at here, it's always marked. In this earlier picture you can see these are all marked 13, 14, 15, 16. This marking is nothing but the presence of the Hox gene. Similar marking is also seen here in the vertebral column, as you can see. So this mark, if you look at here, from the Hox gene 9, the proteins they will make the scapula, from 10 humerus, 11 ulna and radius or radio-ulna together, and from 12 metacarpal, from 13 digits of our finger, or all these things will be produced. So you can see here, if the presence of Hox A and D are there, they will going to form forelimb, Hox C and D, hindlimb. So this is how the presence of all these Hox genes and their associated proteins will signify what kind of structures a vertebrate arm is going to have. Okay, so I hope you understand how the vertebral limb development is done. If you like this video, please hit the like button, share this video with your friends, and stay tuned to watch more and more videos like that. Thank you.